Hello, class. So I just want to go through this DNA genetic code practice I put up on D2L for you. So we're going to go through and review how to do it. So you've got a few sections of DNA here, coding strands and non-coding strands, and then you've got the genetic code, a table of it here. So I'm going to ask you a question on your exam that'll be, here's a strand of DNA, tell me what the mRNA will be, and then tell me what the protein will be. So this will make sure you know how to do it. You don't have to memorize the genetic code. You can have a copy of this with you when you are taking the exam. I think it would be really smart to have a copy of it with you when you take the exam. So let's start here. Number one, our coding strand is ATG, AAT, CCG, TGA. And our non-coding strand is TAC, TTA, GGC, ACT. So if you are RNA polymerase, you are going to come in and this non-coding strand is going to be the template strand. That's what you're going to bind to. And so you'll say T binds with A, so you'll put A. A binds with, well, it binds with T in DNA, but in U and RNA you put U, and C would bind with G. Then you say TTA, so T would go to A, T would go to A would go to U. Then GGC, the G would bind with C, the G would bind with C, the C would bind with G. Then ACT, the A, well, normally it would bind with T, but in RNA it's U. The C will bind with G, and the T will bind with A. So that's how RNA polymerase is doing it. But if you are a student on a test, there is a shortcut you can take. You can take this coding strand and just copy it, but turn the T's to U's. So the ATG, we turn that T to a U, ATG turns to AUG. AAT turns to AAU. CCG, CCG. TGA, UGA. Okay, so that's all you have to do. You don't have to think, what is RNA polymerase going to bind? Like, you know how it's going to bind to the template strand and base pair everything. But if you're a student, you can take that shortcut and say, copy the DNA, the coding strand, turn the T's to U's, okay? So now you have your mRNA. Then to figure out what your protein is going to be, you're going to come over here to the genetic code chart. So we start with AUG. Remember, our codons are 3 nucleotides at a time. Those 3 nucleotides in one codon will code for 1 amino acid. So we come over, A is the first one, so we come down here, here's the first position, A. Then second position is U, so second position is U. So AUs are all here, and then AUG is methionine. Now we know methionine is always the first codon. Or methionine is always the first amino acid. AUG is a start codon. So if you are doing something and it doesn't start with methionine, you need to back up. Something went wrong. And if your mRNA doesn't start with AUG, you need to back up. Something didn't go right. Okay, so we have methionine first, then AAU. So we come to A, then across AA, and then AAU. AAU goes to asparagine. So we'll just write on ASN. Oof, don't autocorrect on me. Okay, then we're going to come down. The next one is CCG. So we're still going 3 amino acids, 3 nucleotides at a time for 1 amino acid. So that's CCG. We come down C, then C is the second, and G. CCG is going to be proline for the next amino acid. And then UGA. So U here is the first position, then over to G. UGA is our stop codon, so we're not going to have anything there. The stop codon just tells the ribosomes to release, everything's done. So our protein there, I would write it as ASN PRO. Okay, so that's all you have to do. So pause it, see if you can figure out number 2, and then come back and I'll go through number 2 with you. We'll see if you got it right, okay? Okay, you ready to work on number 2? So we're going to take the biology student shortcut and just take our coding strand and turn the T's to U's. So ATG, AUG, CCT, CGU, GCC, GCC, CAG, CAG, AGA, AGA, CTA, CUA. So copy your coding strand, turn the T's to U's. For our protein, AUG, that's a good sign. We know our first codon should always be AUG. And then AUG, AUG, that's methionine. Okay, then what's our next codon? CGU. So we come C, G, U, arginine. So we're going to put ARG. Then GCC. We come over here. G, C, C. GCC is alanine. Okay, I'm trying to keep the proteins lined, the amino acids lined up under the codons. CAG. So C, here's our first position. Come over to CA for our second position, and G. So CAG is glutamine. AGA is our next codon. A, G, AGA, arginine. And then CUA, CUA, leucine. So that amino acid sequence would continue on until you got to a stop codon. Okay? When you're doing this, pay attention and see how these codons both give you phenylalanine. These two codons both give you leucine. All of these codons give you leucine. So that's because the genetic code is degenerate. Remember that from the lecture? So that gives you a little bit of room for your DNA to have a little bit of mistakes, or for your mRNA to have a little bit of mistakes without ruining your protein. So it's a little bit, a nice little bit of wiggle room that your cells have there. Okay, so go ahead and work out number 3 and number 4 and see if we can come back and work it out together. Okay, you ready? So, coding strand. We're just going to take the coding strand, turn the T's to U's to make our mRNA. So ATG becomes AUG, CGT becomes CGU, GCC stays GCC, CAG, CAG, AGA, and CTA becomes CUA. Okay, so we just took this coding strand, and that's the same as this strand, except we turned the T's into U's. Okay, DNA has T's where RNA has U's, so we don't have the T's in RNA. Okay, and then AUG, we know we've got the first part right, at least. It starts with AUG. Okay, so the protein for AUG, we say the first position is A, second position is U. AUG is methionine. Always starts with methionine. Then CGU, we come over here. Here's the first position C. Here's the second position G. CGU gives you arginine. GCC, we come here, first position G. C in the second position, so move over here. GCC gives you alanine. CAG is our next codon. Those 3 amino acids that code for one are the 3 nucleotides. Someday I'll say that right. CAG are the 3 nucleotides that code for this next amino acid. So we say C in the first position, okay? A in the second position, CA, and then here's CAG, glutamine. Okay, then AGA is our next codon, our next 3 nucleotides. A in the first position, G in the second position, and here's AGA right there, arginine again. And then CUA, C in the first position, U in the second position, and then CUA is leucine. Okay? Okay, did you get that right? If If you need to go back up and do it, if I make a mistake while I'm doing this, let me know and I'll fix it for you. Let's go ahead and do number 4. Try to do it on your own. Ooh, my font suddenly changed. Okay, so we're going to take the coding strand and take that strand of nucleotides, and we're just going to copy it, except every T becomes a U. So ATG becomes AUG, TCA becomes UCA, GAC becomes GAC, GGA will be GGA, TGC will be UGC, and TTG will be UUG. Okay? So that's our mRNA. Just this coding strand that we turn the T's into U's. ATG, AUG. Okay? So now for our protein. Oh, didn't want to do that. Okay. So let's go and look at what our protein will be. So AUG is our first codon. So A, U in second position, AUG. That gives us methionine. So we're happy because right away we started off with the start codon. Okay, don't ever pick an answer for mRNA that doesn't start with AUG. Don't ever pick an answer for the protein that doesn't start with methionine. It always starts with AUG. It always starts with methionine. Okay, next is UCA. So we come to U in the first position, C in the second position. UCA gives us serine. So our next amino acid is serine. Then GAC. So GAC is our codon, our 3 nucleotides that are going to code for 1 amino acid. So we come down and find G in the first position, A and C. GAC gives us aspartic acid. So we say ASP. Then GGA, we come over here, G for the first position, G for the second position. GGA is glycine. Then UGC is our next codon, our next 3 nucleotides. So we say U, and we say G, and we say C, so cysteine. And then UUG. So UUG gives us leucine. So when you're doing this, you can see how easily that if you put in 1 nucleotide or took out 1 nucleotide, like if you put in a nucleotide here, if you put in an extra A here, how that would shift your entire coding sequence down to be, you would have an entirely new sequence of nucleotides. You can see how that would work by just sending in 1 nucleotide. You have changed all your codons. So now your codings, you would have an entirely new mRNA, or you'd have a similar mRNA that would give you an entirely different protein because your codons have changed completely, even though you only put in or took out 1 nucleotide. Okay, so I hope that makes sense to you. There is a lot of coding practice on the internet. If you just look up for the genetic code, how to make mRNAs, how to make proteins, how to use the genetic code, there's a lot of information there. So don't hesitate to get out there and practice using this, because I will ask you, I will tell you, here's a strand of DNA. What is the mRNA going to be? What is a protein going to be? You do need to be able to do this. You need to be able to tell me what a codon is, how many nucleotides are in a codon, how many amino acids a codon codes for. You need to know that that codon binds to the anticodon on RNA, which is carrying these amino acids. You need to know those types of mutations and how they're going to affect your protein. So make sure you're paying attention and studying those things in Chapter 8. Your exam is coming up pretty quickly. So let me know if I can help with anything, and I'll see you all later.